Welcome to Sports, Clicks, and Politics with your hosts, Ben Hussong and me, Shawn Hannon. All right, let's welcome to the show Chief Science Officer and founder of Medicinal Genomics, Kevin McKernan. Friend of the program, Mr. McKernan, welcome back to Sports, Clicks, and Politics. How have you been?
I've been crazy busy lately. Apparently, a lot of dodging lots of arrows after we published some things people don't like.
Well, I think that's been kind of like maybe your life for the last couple of years, kind of poking holes in some of the mainstream narrative and taking shots back from it.
Yeah, indeed. I was always worried. 11 years ago, we got involved in the cannabis industry, and I was worried about some cartel might take me out, but go sequence a vaccine, and then you'll get the real brunt of the spear.
They are their own cartel, I guess, right?
Yes. Yeah.
So, something with a little bit more teeth with the U.S. government back behind it. So, well, kudos to you for picking fights with the powers that be. It's much appreciated amongst the people here. I promise you. So, I brought you on today just because we had this new article here, and we'll touch on this and maybe find out why it's important here. But this was an article published in the Brownstone Institute. Headline reads, "The Vax Gene Files, an Accidental Discovery." It talks about you and your accidental discovery and bear with me for my lack of technical expertise here, but you called it a plasmid DNA that you found in basically vials sent, unused vials, I'm assuming, of the Pfizer and Moderna vaccines. And you were using these vaccines for something else, apparently, some other sequencing problem that you were trying to find. And when you got into these vials, you found something that you were not expecting. Why don't you tell the folks what you found?
Yeah, so if you look at my history through Twitter, the last few years, I've been somewhat critical of the vaccines for not having had any sequencing information performed on them to understand if they're really what they claim they are and what the error rates are. But I never could justify sequencing them. Despite that, people sent me vials, and I just would shove them in the freezer for a rainy day. And then, one day we had an experiment that it was an RNA sequencing experiment in cannabis. We were trying to figure out what's going on with this viroid. And it wasn't working. We're getting sequencing that looked like it was just all over the genome. It wasn't enriched for genes, which is a sign that your RNA sequencing isn't working. So one of our informatics people said, "Hey, you should spike in some genes that have poly-A's, and then you'll know whether your purification's not working or whether your process is screwed up somewhere else." I said, "All right, I've got some of these things in the freezer. I'll pull them out." Unopened vials, but by that time they had expired. But we threw them in to sort out this experiment, and it worked. It pointed out that we had a DNase problem. But as a result of this, we got really, really deep sequencing over the Pfizer vaccines. And so Pfizer and Moderna, the bivalent vaccines. And so when we start digging through that, we realized their expression plasmid was actually still in the vials. The expression plasmid is how they make these mRNAs. For those not familiar with the process, in order for Pfizer to make, and Moderna to make, trillions of these per shot, they have to have like a mold that stamps out lots of mRNA. And that mold is usually DNA, and it's in the form of a circular plasmid that they replicate in E. coli, so they have lots of the mold. I don't mean like a fungi mold. I mean like, kind of injection molding type of thing. It's just an analogy. But so they take these plasmids and run a reaction that makes mRNA and incorporates their modified nucleotide, and they try to make lots of this. But ideally, they should be getting rid of that. So I was kind of shocked when I found that, because I didn't budget any time to go take on Pfizer. This is just something that came through one of our experiments, but I knew I couldn't just bury it because it was clinically significant. The fact that there was a lot of this DNA in there, people need to know about it. So that put us into a bit of a race to go and verify it, as many different ways as possible, many different technologies we could in our hands, and to publish methods that would allow other people to replicate it, so that this whole process of peer review, which can take a year, can be short-circuited, and other people can just replicate our work by reading a Substack or a preprint and ordering some of the reagents to do it themselves. So that's where we ended up.
Yeah, and so you talked about you were kind of raced against time here, so you kind of publish this out in unconventional methods.
Yeah, we did. We got a lot of heat for that. So, it's a good question. What do you do when you discover a contaminant that's in a shot that's going into billions of arms at a time and getting pushed into pregnant women and into children? You don't want to carelessly yell fire in a movie theater. So the best thing you can do is transparency. We started putting the data up like every 2 or 3 days. We put a subset with what we find. So you would find some mistakes. It would be, okay, this is what we discovered today off using this technology. Tomorrow we're going to use a different tool. The different tool has a different opinion of what we measured with the first tool. We ended up measuring this like 10 different ways. And in the end of the day, I think we ended up using qPCR as being probably the thing that the public understands the most and has been used throughout the pandemic to measure the virus. We figured, well, that's a fair enough tool to use to measure the contaminant in the vaccine. And even with that tool, the vaccines are over the regulatory limit that was set by the EMA and the FDA for how much DNA should be in these things.
So that leads me to the question of why were they not checking for this, or were they checking for this and didn't care, or
that's the good old question. So after we discovered this, we started digging through the EMA documents, and in fact, we weren't the first to discover this. Pfizer was. Pfizer disclosed this to the EMA that they had an 815-fold variance in the amount of DNA contamination they had just across 10 lots, and these are 10 lots that Pfizer got to cherry pick and hand data to the EMA. I'm pretty certain they didn't go and give them their worst ones. So 815-fold variance on a process across 10 vials. If you expand that to 1,000 vials, I guarantee you there's probably 1,000-fold variance or 10,000-fold variance when you start expanding that to more than 10 vials. So the lots that we sequenced, that's only a few of them, and they don't represent any of the lots that were in the Schmeling paper. For those who aren't familiar with the Schmeling paper, these guys published work showing there's a very batch-dependent effect on adverse events with the vaccines, where like the vast majority of the adverse events are coming from a few lots. We're not looking at those lots. We're looking at lots that pretty much have a clean track record, with the exception of the monovalent lots we subsequently looked at as one that's not doing as well in California, according to some work that Sasha put out. But the bivalent ones were lots that didn't necessarily have a high adverse event ratio. So whatever we're finding there is being over the limit. We're very curious to find, well, what are the double-stranded DNA contaminants in the vaccine vials that have some notorious history to them. So yes, the EMA is aware of this. The EMA has I have a Substack that goes through all the language the EMA has thrown at Pfizer, saying that these do not meet GMP standards, that these things are in fact lacking clarification on this particular step that they have to remove the double-stranded DNA. So there's a lot of criticism in the documents about how Pfizer hasn't really satisfied their criteria for getting rid of this contaminant.
And so I guess I think this is probably the question that everybody want to know is what could happen? What are the outcomes of having this unwanted DNA in a vaccine, and can it affect the DNA of a person who was injected with a vial with this contaminant?
So this is where all the spears come out. If you speculate on this, is when everyone starts to attack you. So just let everyone know we're in the room of speculation at this stage. I think the most important topic on the table is there's been a long debate for the last few years as to whether or not this vaccine can integrate into the genome. I think Rudy Ginesh of the Whitehead has shown that the actual virus can. All right, that's settled science now. He was attacked when he first put this out as being an anti-vaxxer. He's not an anti-vaxxer, but he went and doubled down, redid the work, and showed that he can get the virus to integrate. Okay. Now, it wasn't a large piece. It was like 1,400 bases or something, but he still got parts of the virus to end up and demonstrate they ended up in the genome. He did try to do some experiments seeing if the vaccine would do this, but he didn't actually have the vaccine. So he made some model templates to try to emulate it and didn't have much success. But many argue that you really have to repeat it with the actual vaccine because it's a different beast. So the question is, will this vaccine reverse transcribe itself into DNA? And there's a group called in Switzerland, the Alden Lab, or Marcus Alden. I'm sorry, I could have his actual jurisdiction mixed up, but Marcus Alden's the paper to look up, and they showed that the vaccine itself can get reverse transcribed into DNA. That's the first step. If you want to get genome integration, you got to turn the RNA into DNA, and then it can get into the genome. The point of our work is to demonstrate that even that step isn't necessary. Many people were critiquing Marcus's work because he did it in cancer cell lines, like liver cell lines. I think it was, and as a result, they were trying to claim that that's not a good model. Well, his model, as important as it is, isn't necessary if there's DNA in the background of the shots, because you don't need a reverse transcription step. You already are delivering the DNA. Now, the DNA that we've measured to date is very short in length. We don't have long pieces of DNA. We've done a couple nanopore runs on this, and we're not seeing anything longer than 1,400 bases. We've been trying to do some transformation experiments as well to see if it'll actually stick inside of a coli and grow, but those haven't been very productive. So right now, most of the DNA that we're seeing in the lots that we have is fragmented in small pieces of DNA. But fragmented DNA is still a concern for the FDA. They specifically point this stuff out. Linear DNA is more likely to integrate into a genome than circular DNA. So they're still concerned that this could happen. Now, the other thing that was unique to the Pfizer vaccines was that they had a promoter in their vaccine that was that they hid from the EMA. There's a little piece of DNA in there known as an SV40 promoter. Now, SV40 has a notorious history in the vaccine industry. It contaminated the polio vaccines, but that was the entire 5,000 letter virus. This is just a short, like 340 base pair promoter. Now, this promoter turns on gene expression. So if it, for any reason, integrates into the human genome in front of a gene, it will turn on the expression of that gene. If it happens to be an oncogene, then you've got problems. So there has been concern that this SV40 piece of DNA is in the Pfizer vials, and if that, for any reason, gets integrated into the genome, it will turn on gene expression of wherever it lands. It's likely to be a very rare event if it happens, but if it happens in a stem cell, which proliferates into many other cells, you've got an amplifiable rare event. So we've seen some evidence through these biodistribution studies that these LNPs don't stay in the muscle. They go everywhere, which means we've got to be concerned about it getting to sperm cells, to oocytes, eggs, and getting to any sort of stem cell with LNPs. Now, what some of this is showcased in the preprint we put live, but what did not make it into the preprint, because it happened subsequently to the preprint being submitted, is that we did some work after we got trolled by someone funded by the Wellcome Trust. They claimed none of this DNA was going to be packaged and it would just be eaten up in your bloodstream. We actually did some experiments to show that no, it's in fact packaged. It is inside the LNPs, and it will be transfected into any cell that those LNPs find. So you'll be getting both the Pfizer or Moderna mRNA, and the contaminating DNA is going to be delivered into a cell. Will it integrate? We don't know. Have the integration risk gone up based on this data? Absolutely. The argument that they've thrown at Marcus Alden and Rudy Ganesh in the past is now off the table, because we have DNA in every shot. And the question is, at what frequency does this integrate, and does it drive any oncogenesis?
And so the LNP, the liquid lipid nanoparticles, right? So if I'm going back to during some of this vaccine discussion, I think those were being found in the reproductive systems of females, and among other things, right? Oh
yeah, there's issues with sperm motility has been going down. I think Arnie Burkhardt, who unfortunately just passed away, was doing a lot of pathology work on this, demonstrating spike protein contamination in a variety of tissues. So it's in. They're also finding the mRNA in breast milk and spike protein in breast milk. So there's a longevity problem on these, and that problem just got worse because DNA degrades more slowly than RNA, and so they're injecting DNA. They've got a problem now. The pieces of DNA probably aren't long enough for them to be expressed into proteins, at least in the lots we surveyed. The argument that's been thrown back at us is that, well, you guys looked at it clean lots, go look at some dirty lots. I bet the DNA is worse, particularly if the EMA has Pfizer on record with an 815-fold variance in how that process behaves. So the verdict's still out. We need to survey more vials, and that's one thing that we want to get the word out about. If there's anybody out there that has PCR instrumentation and access to these things, we've put all of these protocols public for anyone to just take a millionth of a liter, which is a microliter, and you can get a CT of 20 off of this in PCR. And I think more people want to see that reproduced, to know that we haven't just put up some type of fear campaign here. But 1 microliter is 1/300th of a dose, and that gives you a CT of around 20. So if people remember, they were being quarantined for CT sometimes at 35, maybe 40 for the virus, and they were surveying that outside of your mucosa on your nose. Most of that was probably subgenomic RNA, not replication competent RNA. Now we have a contaminant that's being injected past your mucosa, so your defenses, and it is at a CT 1 million times higher. and no one cares. Well, I mean, some of us care, but let's just say everyone else is trying to brush it off as like, oh, well, maybe it's contaminated. This is what we're hearing now from the proponents who have attacked our work is, fine, maybe you've convinced us it's contaminated, but it doesn't matter. Those amounts are minuscule. Well, those amounts were not minuscule 2 or 3 years ago when they're testing everybody for COVID. Now that they're injecting them past your mucosa defenses, they're suddenly immaterial. I think that's a bit of a double standard that they're standing on.
Yeah, and that's kind of what I was going to go. It's like, what are the the people who are trolling you or pushing back? Like, what is that their main argument? Is that, okay, yeah, you got it, they're contaminated, but so what? And this.
Yeah, they're most of their arguments are taken straight from our own paper. I mean, we list these things that are limitations of the study, which is that we're not in control of the provenance of these files. We got shipped them anonymously, but they were sealed, so they didn't look like they were tampered with. But maybe they baked them in the sun for a few days before they sent them, and the RNA just got degraded. We found the RNA. We were able to sequence the RNA, so it's not gone, but it may be lower in quantity than they initially estimated. In fact, there should have been 30 nanograms in there, and I think in some of the vials we're finding about half that. And that 50% loss is probably an artifact of us purifying the RNA out of the LNPs. We lost them in the process. I don't think they were heavily degraded. We did run them on Agilent tape stations, and we didn't see heavy signs of degradation. We saw the typical. I don't know if you're familiar with this Blotgate story that's been out there, but when you look at these Agilent traces, you can see some of the RNA is not full length. And this is something the EMA cited and lowered the standard for them. They wanted them to have 80% RNA integrity. When Pfizer couldn't hit that, they lowered it to 50. All right. So we ran these things on those machines, and they didn't look too different from what they've published. So we don't think they were overly degraded because we've got data showing that the RNA integrity is in fact intact. And then second to that, you can't really describe that if someone were to cook these in the sun or mistreat them, how a plasmid would suddenly spontaneously emerge inside of the vials. And that plasmid happens to have sequence that maps the map that they gave the EMA. Of course, the map that they gave the EMA, they erased the SV40 promoter from it and gave them everything but that. So they clearly don't like that thing and don't want people talking about it. So it's something that they've hidden due to the fact that it has a notorious history in the vaccine field.
Yeah, and it seems that there's been enough clamoring about the safety of these that they're going to try to hide anything that might give them a red flag, I guess. Is that a fair assessment of what you think why they're hiding that promoter?
I think so. Yeah, it's there's no reason for them to pull it off. They put every other part of the plasmid in there. They put the F1 origin in there. They put the cannabinoid resistance gene. They put the spike protein in there. They put the EMA 1104I cut site in there. But they just omitted that one piece, which is the controversial piece.
Okay, and so what happens next? Obviously, you put this information out, allowed for, you said people can, anybody who has PCR capabilities could replicate your trial there, or your.
Yes, some of that has happened. So we've sent kits out all over the world. We're always eager to send out more to other people. And if you don't trust us to make them, you can order we've put all the recipe online. So you can order your own oligos from like a provider like IDT if you need to, and make your own primers. We've also put all the sequencing public for the vaccine, the plasmids we found. So if you don't like the primers we design, you can design your own. All of this is transparent. It's online. Anyone can download the data and view for themselves. So a group in Japan took our data, our sequencing data, reassembled it, and found the same thing. Another group in Europe sequenced the monovalent vaccines, forwarded us the data. We assembled it and found the SV40 promoter in the Pfizer monovalents from their data as well. So there's been some replication there. There's been some replication in Japan. Another group in Japan did work qPCRing this with a slightly different PCR assay using CyberGreen, but got similar CT scores. So it's been replicated about, at least that was on Moderna, on 2 different places in Japan, once in Europe. A fourth person, Didier Riault, who's under a bit of criticism right now. There's a witch hunt after this guy for doing a hydroxychloroquine trial, but their lab knows how to do PCR. They put some of the earliest PCR data out comparing plating viruses with PCR. There's a great paper from Rita Jafar on this. They claim to have found DNA in the vials as well. I'm missing somebody. There's another group that's also had some preliminary results on this, but it's starting to emerge in multiple labs. They're finding the DNA. The real question is how much and how much does it vary lot to lot? And then the third question emerges. Whenever you see plasmid DNA in any injectable, it rings alarm bells that you need to look for the E. coli endotoxin that's probably present with it. So these plasmids are grown in E. coli, and you have to crack open E. coli to get the plasmids out. And that process exposes all of these surface glycoproteins, lipopolysaccharides, if you will, to the solution. And if any of these endotoxins get in, they can create anaphylactic shock. Many of the symptoms you find of people like dropping right after vaccination, it's probably an endotoxin that's doing that. And whenever we see plasmids, that's the first thing we think of is, okay, there could be endotoxin in here. If there's endotoxin getting injected, those numbers need to be revisited, because if that varies lot to lot, then we got a disaster on our hands that could be explaining a lot of the adverse events that are out there. So the third thing I'll mention is that there are papers out there on our Substack that will show that if you inject enough double-stranded DNA alone, it's prothrombotic. So we already have evidence of thrombosis after these vaccines. So here we have a contaminant with literature that links it to one of the adverse events, and people are trying to bury it. Now, whether it's it could be a spike protein that's causing the thrombosis, but throwing in more thrombotic agents into the mix as contaminants is something that it's a very fixable problem. That's what people need to know, is that this is not something that necessarily damns the whole platform. They can fix this. It's not expensive to fix this. This is just them going too quickly and not dotting all the I's and crossing all the T's in QC. They could probably fix this for under $5 million. It's just they need a new enzyme. They need to tweak that process that they have to get rid of this DNA, and they'd have cleaner mRNA. But that's not getting done. I think what you're going to find is it's probably cheaper for them to like change the regs than it is to actually clean it up.
Do we think Pfizer has an extra 5 million laying around or no?
I don't know. They seem to be having a really hard financial time lately.
All right. So what? So I don't know if this is even related or not. This is just where my brain was. So is any of this like I have my haven't figured out what to make of long COVID, but is any of this relatable to that? Like, I don't know.
It's possible. I mean, that's what people. Yeah, that's what people are bringing up, is that, okay, maybe the DNA or the RNA that we've been detecting so there's some papers showing they've detected the genome from the vaccine 28 days later in people's plasma. Then there's the data showing some of the stuff in breast milk. Those studies are using tools called RT-PCR, which amplify DNA and RNA. So it's possible they got amplification off the DNA, not the RNA. So those might need to be revisited based on the methods. But yeah, now that we have a DNA molecule that's in there, the longevity of this and how long it's detectable in the blood is up for debate. So we have been doing some work designing an assay where you can use this. The assay that we designed was meant for targeting the vaccines. So it has two different assays. It targets the plasmid and it targets the spike protein. And you can run that with quantitative PCR, which only amplifies DNA, or RT-qPCR, which amplifies both RNA and DNA. What we're adding in is a third component that will pick up something known as RNase P, which is a human gene. So if you want to apply this on human tissue and look for this type of contaminant in a human biopsy, you'll want that internal control to show that your tissue's there and you can amplify the tissue and the RNA prep from the tissue behaves. So we're making a third assay. So the thing that's used to detect the contamination of the vaccine can be turned and looked at human tissue to see if you can find it in patients. And that might help a variety of pathologists try to answer the questions that you asked, which is this what's floating around causing problems in detection months later? Is this playing a role in any of this reverse transcriptase activity and a lot of the questions we have about integration? And likewise, the assay that picks up the vaccine is important because you need to be able to differentiate the virus from the vaccine, which can be done with DNA primers. It's a little bit harder to do with ELISAs and a lot of the tools people are using right now. Right now, I think that the main way to differentiate whether someone's affected with long vax or long COVID is to look for nucleocapsid. Nucleocapsid is something that comes through the virus, doesn't come from the vaccine. So if they have nucleocapsid, you can say, aha, it's long COVID, it's not long vaccine. Reality is, based on the vaccination rates, it's probably a little bit of both. But anyway, nucleocapsid has been critiqued in that they don't find expression of it in some tissues. So you can make false assumptions that it's the vaccine and not the virus in some tissues if you sample the wrong tissues. So targeting the DNA or the RNA doesn't make those mistakes. we can readily differentiate these things because the sequence of the vaccines is so different than the virus. They did this codon optimization that made it very different. That makes it very easy for us to target the vaccine RNA very specifically versus the virus's RNA. So having these tools will be very helpful to for the pathologists out there to sort of cut through this quagmire we have of how much of this is long vax versus long COVID.
All right, I have a couple more maybe related questions, I guess. But before we move to those, is there something specifically about this contamination issue that we haven't covered here in the last 25 minutes?
Well, yeah, so also in the realm of speculation, it's something we have de-emphasized in a preprint, but many people raised as being a concern. And early on, we were worried about this as well. But as we got more and more data off of the vaccines we had, we started to whittle away at this being a risk for the vaccine vials that we have. If there are any full-length intact plasmids in these vials, which there may be in some of the ones Schmeling described, or in other vials that have higher contamination rates than what we have measured, if those are in fact intact, those things can replicate inside of the coli or inside of a lot of bacteria. So in this case, the injection would deliver plasmids. They might get transformed into the bacteria in your body, in which case those bacteria would then be replicating that spike protein encoded plasmid. Now, we don't think they're going to express spike protein in the bacteria because the RNA that's inside this plasmid has a human recognition for translation. So it shouldn't translate. This is something known as a Kozak consensus sequence. It shouldn't translate in bacteria. It only translates in humans. But some human cells will go through a process of bacteriophagation where they engulf a bacteria to kill it and then break it open. And then the plasmid could be in a mammalian cell and actually make spike protein that way. So there are some concerns that if the plasmids are around, there could be another pathway for prolonged expression of spike protein in humans. And that would also come alongside with an antibiotic resistance gene, which could cause some other issues. So that's a little bit more remote. We need more data to actually confirm that speculation. But once you start seeing fragments of the plasmid present, the next thing you have to start asking is how many vials have full-length intact plasmids, because that's a whole other risk profile that's not being considered in this. The second thing to be considered is that the EMA, when they wrote these regulations of how much DNA is allowable in these injections, they were not contemplating what if they're packaged in lipid nanoparticles. They just wrote them as, what if they're naked DNA getting into your bloodstream, what's going to happen? You'll get an interferon response. So you have to have this lower than a certain level so you don't have an interferon response. They're also worried about integration. The FDA, Keith Pettin has great work showing that they're concerned about genome integration with double-stranded DNA injections. What they didn't consider when they were writing those regulations was that in the future, there might be lipid nanoparticles that package this stuff so that it can't be destroyed and gets directly delivered to your cells and even maybe your nucleus. And so they probably need to reconsider the regs for LNP delivery. And if there's any replication competent DNA in there, like a plasmid that can amplify on its own, because if you have something that's amplifiable, a small amount, you only need a very small amount of it for it to take off. So they wrote these regs considering naked DNA that might be coming from a host cell like E. coli or from a mammalian cell line that they grew something in. They were not considering these types of DNA elements that are highly transfection competent or highly replicatable on their own. And those things probably need to be reconsidered when people are looking into these injectables.
All right. So I had thought that the COVID and the vaccine was going to be kind of washed away in political history, if you will. I don't know if we're going to touch on a couple things here. So, but I don't know if you saw, well, two things. We have the emergence of RFK Jr. in the presidential race, who has a history of vaccine skepticism, to say the least. So my fear, I had a fear that COVID was going to become a non-issue and people were just going to kind of water under the bridge it and not be something that was going to be discussed for future leaders of this country. I think RFK enters into the race is going to kind of keep that there. But I don't know if you saw Trump yesterday announce something to the fact that, I don't know if he's trying to outdo RFK, but he said basically he was going to start the discussion about whether or not vaccines were safe in like a general sense, not just the COVID vaccines, but all COVID vaccines.
Oh, that's a shocking pivot considering he was doing victory laps over the whole Warp Speed program.
I don't think anybody would disagree there. If he could have figured out a way to sign every single syringe with a gold signature. He would have put his name on every single one of them if he could have. So this is definitely a pivot. And, he didn't mention the COVID vaccines in this statement. It was a video statement he put out yesterday, but he mentioned all the other vaccines and basically
the ones that have been used for decades have been tested. Yeah, right. I mean, I'm not going to defend all of them in the past. I certainly have raised my skepticism ever seeing, when you see how the sausage was made on COVID, you want to question everything else. But at least from an adverse event standpoint, nothing measures up to like these COVID vaccines have more adverse events than all the other ones in history. So of course, if he's going to pivot, he pivots to where the ball isn't going to be. But I mean, maybe he has to because DeSantis is on this train as well. The only person who isn't on this train is Biden, who I'm not certain is going to be intellectually there when it happens. So yeah, I didn't know that. That's a crazy pivot to hear.
That's exactly what I thought too. I was like, this is a like so crazy of a pivot. No, and like I said, he mentioned nothing about the COVID vaccines in this video. He mentioned about every other possible scenario that could be. But I guess my question is can we still get the information if in fact these vaccines, which as you pointed out, the adverse event profile is higher than anything else that we've seen, I don't know that information has actually made it to the public. I guess the reason why I was bringing this up is there stuff still to be learned from these COVID vaccines? And is it possible that politics will keep the fire stoked enough to actually get this information where people are still interested in it? Like, I feel like a lot of people have kind of like just moved on past this, and I can't. So I'm trying to figure out, will the public, will this become public knowledge as opposed to or common knowledge, I guess, as opposed to
right?
So I'm hopeful that the fact that RFK is in the race and Trump just made this crazy statement yesterday, that maybe we'll still get some additional, looking into from people like yourself, with people who are highly credentialed in this, and try to figure out if there is actually something that can be done. I mean, I think the uptake of these vaccines has kind of shown that the people are worn out on it.
Yeah, absolutely.
The powers that be are not.
So I think two things are going to weigh into this. One, this is a platform they plan to use for everything else. So they're not going to stop, whether it comes to veterinary medicine, whether it comes to cancer, whether it comes to RSV. They're going to keep making these LNPs and these mRNAs. So I think we still have to stay on the case for this, because I don't think they've solved these problems. Like, even if they get rid of all the DNA, I don't think they're going to be safe. I don't think that's the major contaminant. As much as it's getting a lot of attention and it's put my name in the news, it's just to demonstrate that they're cutting corners in manufacturing. They're going to switch from a spike protein to like nucleocapsid, and everyone will be like, okay, fine, you fixed it. I don't think that alone is going to fix it either. I think nucleocapsid, when you get in there, it has other, prion-like domains in it. It's got, if you just decorate cells with foreign peptides like this, the immune system goes and clears them out. And so if you do this at a very high density across your epithelium, you get lots of scarring and you get lots of leakage in a lot of these barriers in your body. So they've not run vehicle controls. Like, if you just inject people with the LNPs, what the hell happens? All right. If that alone creates enough cytotoxicity and cell death that you get a lot of these adverse events, then the entire platform is damned. It's somewhat damned out of the gate when you think that when they're going after respiratory viruses through an injection, right? They need, your respiratory viruses are put off by mucosal antibodies. You don't get those from injection, right? So this is something that the entire platform has kind of, done this, these aren't the droids you're looking for kind of crap, where they're injecting you with something saying, hey, this will give you immunity in your nasal mucosa. It doesn't. You have to have something that actually presents to your nasal mucosa for that to happen. So, maybe those nasal sprays that people were doing would be a better route of vaccination. So I think we have to keep the fire on them, because they're not stopping. They have a huge pipeline of these things, and they haven't yet proven that the LNPs themselves are safe. Yeah, and I think what you're going to see them do is goalpost shift on this, saying, oh, look, we got away from toxic spike now, and we got rid of the DNA, and everything's fine and dandy. Let's continue injecting all the cattle with this stuff. On the flip side of this, mainstream media, who's completely bought and sold by pharma, is dying, and they don't matter anymore. And someone like Joe Rogan can have Peter McCullough on and get 50 million views. And I think I heard, what did Tucker get yesterday? I mean, Tucker used to get 4 million at Fox.
Yeah.
You know, right? CNN gets 300,000. I mean, Joe Rogan is 100 CNNs with Peter McCullough.
Yeah.
So as much as pharma is suppressing this, they're suppressing it to the media that's reaching to people that's 60 and older. And those are soon to not be the decision makers anymore. All right. So the younger generation is listening to Joe Rogan, yourself, and in Twitter, right? They're not paying attention to the bought and sold dinosaur legacy media. So I have a feeling it's going to persist for those two reasons. That the media that's out there, and even though it's being censored, it's reaching, people want it. That has it on its radar, front and center. And then they're not going to stop trying to make these things, or can continue to bring them to market with some of the same technological flaws. That the argument's going to persist for another 5 years, if not 10.
And correct me if I'm wrong, but wasn't the lipid nanoparticles the main problem of mRNA vaccines the whole history of them? They weren't able to actually find a safe lipid nanoparticle, and then miraculously they found one right before they needed the COVID vaccine.
It could be. I'm not as well read on the history of the LNPs. I just noticed in the Pfizer trials, they never ran that. Their control was a saline injection. It wasn't an injection with LNPs. So that vehicle control is missing. And Mark Giardo does some good work on this. He's published on COVID Mythbusters, where he goes through with just the impact of transfecting this many cells with these LNPs can create immune attacks against a large portion of your epithelium and create all types of havoc. And some of this he's even tied back to previous vaccination programs that don't have LNPs demonstrating there's risk of driving immune responses if any of these things just hit the circulatory system and don't stay in the deltoid. What can happen? And so I think the whole administration of these things needs to be revisited so that they're not taking on these risks throughout vaccination.
All right, before I let you go, I just want to give you one last chance to make sure we touched on everything that you would want to touch on here. Vaccines or not vaccines, if there's something else you think that should be put out there, I'm all ears.
Well, I think if you circulate our preprint to anyone, the first thing they're going to hit you with is it's not peer reviewed. I'd push back on those people saying peer review is meaningless. What you need is reproduction in science. Having 3 people sit in a room and critique your paper when you don't know who they are or what conflicts they possess and how much money the pharmaceutical company is actually driving through advertisement to that journal, that's a church. That's not science. Okay, that's a set of clergy that have financial interest deciding what gets published. What matters is if people can reproduce this work. So pay attention to who reproduces it and when. And if you want, chip in and reproduce it yourself or tell us we're wrong. We've put everything public so anyone can scrutinize that. This peer review process, which we'll probably go through tortuously, but it will take probably 6 months to a year. We're already getting data back from labs that have run this PCR reaction before then. So I think before that process even puts its boots on, we're going to have 5 to 10 labs that have reproduced this, and the peer review is going to be ceremonial. So reproduction is what matters, not this peer review process that has been corrupted through the entire pandemic process. But just note that that's the state of where it is. We don't have evidence of genome integration. We have evidence of this SV40 promoter. It's not the whole virus. We don't have strong evidence the plasmids are intact, but there's a risk that they could be in some vials. So it's all very preliminary. However, none of this should be there. And the regulators knew this, and they warned Pfizer against it, and they injected them into people anyway. The most stunning thing you'll find in the EMA documents is that they injected people with the contaminated lots. They knew they were contaminated, injected them, and then they removed those people from the preliminary analysis in the trial. So people in the trial got some of these lots, and the data didn't look good, so they pulled them out. Now, the trial was done on lots that didn't have this problem. And then after the trial was completed, they switched to what is known as process 2, where they made these things in E. coli, and the problem existed. Right, so there's a huge bait and switch going on in the Pfizer trial. They did this on synthetic DNA and synthetic RNA that was generated in vitro for the trial, and then they switched to this E. coli thing after the trial was approved and gave somebody these injections that were not the same as the ones that were used in the trial. So dig into the trial and dig into the EMA docs. You'll see that this process 1 versus process 2 is where all the shenanigans go on in the trial. and it has to do with the DNA contamination.
All right. Well, Mr. McKernan, I thank you for your time. I thank you for your volunteer research here, keeping us all informed on what the crazy pharmaceutical companies are doing to us. Maybe the next time we have you on, we'll talk about something maybe more uplifting, but just as
yeah, sure. I actually work in a much more uplifting field, in the cannabis field. So we can talk about how to, basically, do an end run around the FDA and the pharmaceutical industry by growing your own medicine. That's actually where our passion lies. So if you want to see any of that, you can follow us at Medicinal Genomics, or CanMed is a conference we run that shows. It's a scientific conference about how you can grow your own medicine and what you need to do in order to make sure that it's safe. So this is something we want to decentralize. We can't be having medicine get centralized through agencies that are purchasable. We got to have a movement toward understanding your own medicine.
And if I'm real quickly, you're doing the psilocybin too, right? Not just the cannabis.
We are, yeah. We sequence about 120 of those genomes as well, and that's a very powerful and exciting field. Those compounds have a remarkable therapeutic index. There aren't overdoses with these things. They are showing a real promise in treatment-resistant depression and PTSD. So
addictions, yeah.
I have a feeling there may even be some implications in COVID, but that's a bit of a stretch. And there's serotonin biology with fluvoxamine in COVID that I think psilocybin will probably emulate, but there's no study that's been done on that. It's just a hypothesis at the moment. So yeah, there's a lot of compounds that we as citizens they're currently illegal. They're becoming less illegal over time. That we need to put some guardrails around, bring some science in, and see if we can clean up the testing so that we know how to analyze and quantitate these things.
Yeah, that sounds like a good future next interview for you. So on that note, I'll let everybody go today. Mr. McKernan, thank you for your time. Again, thank you for your service, and we will hit you up again in the near future.
All right, thanks, Shawn. Always a pleasure talking to you.
Always, thank you.
All right, take care.
Take care.